DNA-encoded library, or DELL, is a technology that enables fast screening of billions of small molecule compounds for drug discovery. This is a DELL molecule. It contains a chemical compound with a DNA label. Thanks to sequencing technology, the compound can be conveniently identified, like scanning a barcode. By applying combinatorial chemistry, building blocks are conjugated to establish compounds. Connected with DNA labels, compounds can be uniquely identified. Let's say there are 1,000 different building blocks in each round. If you perform 3 rounds, which is 1,000 cubed, 1 billion different combinations will be achieved. DELL enables mass creation of chemical libraries, increasing the chance of discovering hits. Conjugation with DNA labels significantly accelerates the screening process. DNA-encoded library is a groundbreaking technology. Screening billions of compounds can be accomplished in one small tube. Minimum amount of target proteins is needed for incubation with billions of DELL compounds. Compounds of high affinity are attracted by target proteins and bind to them. Those that don't bind to proteins are washed away. By shifting elution conditions, strong binders are isolated and collected. The isolated binders are decoded by reading DNA labels to access their chemical structures. In contrast to traditional technology, DELL no longer requires separate experiment for each compound. Instead, experiments are performed in a single tube. DELL has reinvented hit discovery process and is able to deliver screening results in 6 to 8 weeks. The efficiency of drug discovery is improved when time is saved.
All right, thank you for that. I'm Arslan Arif, the founder of Endpoints News, and we're here today for a DELL Kits special event, a new method of hit discovery. Let's move to the presentation. It is my pleasure to introduce Alex Satz, who is a Senior Director of DELL Strategy and Operations at WuXi AppTec. I want to remind the audience, if you have any questions for Alex, and he is a great presenter, please get those in right now using the Q&A box below, and I will pose those questions to Alex at the end of our session. But without further ado, I now give you Alex Satz.
Okay, thank you. So I will go ahead and share my screen and just tell me if it's showing up properly as it should. Perfect, and I'm gonna enlarge it to fit my entire monitor, and let me get my pointer working. So yeah, my name is Alex Satz, and I've been working in the DELL field now for almost 15 years. And I will go ahead and jump straight in because I am one of 3 speakers. Although my presentation is the longest, it will take about 20 minutes. And the topic here is WuXi self-service DELLs. And what I want to start off talking about is why we decided to make these DELLs and what the major advantages of them are compared to a full service DELL where you send target to us and we screen it. So in the case of the self-service DELL, we create a DELL kit and we send the kit to you, and then you use that kit to actually conduct the screen in your own lab. And the benefit of that is twofold. So the first one is that it allows us to potentially lower the price of doing the DELL screen quite a bit. And what that means for academic users is that, and I'm going to talk about this on the next slide, is that we have set up a way for you to run a DELL screen at no cost, or basically no cost. The second thing is that because the screen is done in your lab, this means that the target that you screen, you don't have to share it with us or anyone else. Also, it means that if you develop new and novel ways to do a DELL screen, you can keep those methods a secret in your own lab, and you can only release those methods when you would like, whether it be in a paper or in a patent, or you could never tell anyone else your secret ways of doing these screens. So those are the 2 main nice things about these DELL self-service kits. So on this slide, I'm going to talk about 2 different kinds of kits that we have. We have the DELL Open Kit and we have the Delight Kit. So let me start off with the DELL Open Kit first. And the first thing is that the DELL Open Kit is free if you're an academic lab. There are a few costs related to shipping things back and forth, but otherwise it's completely free. And if you look at the blue circles here, this represents tasks that we at WuXi do, and the yellow circle represents the tasks that you do in your lab. So for instance, it's our job to create this kit and to send it to you, but then you actually take this kit and you run the screen yourself in your own lab. You then send the physical wet lab samples back to us, and we do the QC, we do the PCR, we do the NGS, and then we share the metadata of the screen with you. So the metadata is all of the data of the screen, but with the chemical structures being scrubbed out. We then help you choose the 5 best hits from your screen, and you then have the choice of having those hits resynthesized for you. But you don't know what the structures of the hits are yet, so we have to synthesize them for you. We then send these hits to you, where you then test them in order to make sure that these hits are active and that they're doing what you want in the assays that you have. Now, if the hits look good, we share the structures of the hits, and there's no cost, and you are free to take those hits and to file a patent, to sell them, to develop a drug, whatever it is that you would like. And from us on the WuXi side, there is no reach through. So there's no milestones, there's no extra fees, there's nothing else outside of shipping and resynthesizing of these hits. The entire process is completely free. But there is a bit of a catch, and that is DELLopen.org is overseen by an academic group. And it's all about being open, or data sharing. And so in order to get the structures of your hits, you need to share the name of the target that you screened, and you need to agree to have the metadata of your screen placed on the DELL Open website so that other academic groups can go and take a look and possibly work with you on later papers. And so the goal of DELL Open is to publish more, to get more science done, and to share things. And for that, we provide the entire thing free of charge. DELL Lite is a little bit different. It's really set up for if you would like to find hits in order to discover drugs. We specifically make this kit towards the goal of there being potential hits in there, which are going to be drug-like. And you have to pay an upfront fee in order to get this kit. Much like the DELL Open self-service kit, though, you have to physically run the screen in your own lab. Then you send the samples back to us, and we do the PCR, we do the NGS. And once again, we share the metadata with you. But the goal here in sharing the metadata is that we want you to be able to make the choice if you want to pay an additional fee so that we unblind every single hit in that data set. And if you look at the results of your screen and you decide, well, this screen looks good, I want to know what these structures are, then you pay this fee, you unblind all those structures, and it might be hundreds of thousands of structures, and we send the entire data set to you. And at that point in time, you are free with this data set to develop drugs, to write patents, to write papers, whatever it is that you would like to do with that data set and with those hits. And again, there are no reach throughs, there's no milestone fees, there's no strings. Those hits belong to you in the sense that you are free to work on them without paying any other fees. So the DELL Open and the DELL Lite kits, they've only been around for 1 to 2 years. And so we are often asked, how often are they actually used thus far? And the answer is here, we have shipped 346 kits. So this slide is just a fun factoid slide. So every DELL kit that we ship out, it has 4 tubes. And every tube has billions of discrete structures. And when you screen a DELL or the DELLs in those tubes, you effectively do a single point binding assay of every chemical structure in every tube of every DELL kit. And if you do all the math, what that adds up to is so far those 346 DELL kits have translated into almost 300 quadrillion single point assays being done. And this is a number that's just going to get bigger as we produce and we ship out more and more of these kits. We wanted to show you this slide just because it's such an outrageously large number, and yet it is mathematically true. Okay, so of all the kits that we've shipped out, we've also shared almost 17 million different structures from these kits. So that's both good and bad, perhaps. So you might think, well, it's great that so many people unblind the data sets and have been receiving so many potential hits, but you might be concerned that if we've already shared 17 million structures from these kits, that there might be very little good stuff left for you. So all the people who got in early have seen all the good hits, and now when you screen, you won't find brand new stuff. But do not fear. It turns out that there are so many structures in these kits that even though we have shared 17 million structures, that only comes up to 0.008% of the total structures in these kits. So only a very small amount of these kits have actually been shared. And towards the entire purpose of this, we now have a third generation kit. Now, what does that mean? So what's nice about the world of DELL is that a single chemist can make a single DELL in the period of 1 to 3 months. And so one person can make millions of brand new structures that have never been before made and never before been screened. And what we then do to our advantage is that we make new DELLs every year. And now we have a third generation DELL kit containing 50% more DELLs. And what that means is that there are billions of new structures in these DELL kits that have never been synthesized before and never been screened by anyone ever. And all these structures are now contained in this new DELL kit. And if you order this kit and then you screen, whether it's DELL Open or Delight, you'll be able to screen a lot of structures that no one has ever seen and no one has ever screened. So we at WuXi, we have our job to do with this. We send you a kit and you put in the time and effort to do the screen. And even if it's a DELL Open kit and it's free, you still have to commit the time and the effort to actually physically do the screen. So if you were to send the samples back to us and we were to process them in a bad way, then that would be an issue, right? So what we've done is we've created an automatic workstation. And what this allows us to do is assure ourselves and you that every sample that we get, we can process the same exact way, and that we can process over 6,000 samples per year. And what we've done to test this is we've taken 15 or 16 different samples shown here on the x-axis, and we have processed them repeatedly over a span of 5 different days. And all these data points for each repeated process is shown here. We've then sequenced each one of these, and we found the correlation between the sequencing for each one of these, which were processed across 5 different days. We see that on the y-axis, the R squared varies from 0.98 to 0.998. All these samples are tightly clustered, and what it shows is that we can process samples over and over, different people on different days, and the results are always the same. So running a DELL screen for the very first time, so many of you might order a DELL Open kit or a Delight kit, you've never actually done a DELL screen. It's not a hard thing to do, but it can be a little stressful. And the reason why is because you don't know after you've done the screen if it actually worked. And then on top of that, you have to pack it and ship it. And especially during these days, no one ever feels that secure about shipping things and about when it might show up. So we now have a portal that you can sign up and go to, and you can track how the shipping is going. You can see if we have gotten the samples. You can see how your sample QC has gone. You can see how the sequencing is going, and you will know when the data is ready for you to actually view and to make the next step with. And hopefully that will make the entire process a little more smooth and less stressful. So how do you get inside of this portal? Pretty simple. You go and you log on. You put your email here, and if you're like me, you will generate a password, which you won't write down. You won't remember what it is. And the next time you sign up, you'll click here on the forget password, and you'll go through some extra steps, and you'll be able to log in. And once you've done that, you can track the entire series of steps from the samples being shipped until when we are ready to share the metadata with you. So on the portal, you'll be able to do everything that you need, including ordering more DELL Open kits. And in a couple of slides, you'll see why it might be important, because you might need to order more DELL Open kits depending upon how your screen looks. All right, so in the past, what we've always done is that when the screening is finished and the sequencing is done, we create a metadata package on a PDF, and it's a static look at how your screen went. But everyone wants to look at their data in a different way. And so what we've done is we've created an interactive way of you being able to look at your data so that you can make the choice, whether it's a DELL Open kit or a Delight kit, how you want to move forward. So let me just give you an example here. So for instance, let's say you only wanted to look at hits that show up in condition 2, and you don't want to look at hits that also show up in conditions 3 and conditions 4. So you set a threshold of enrichment in condition 2, and you set a threshold so that you say that things don't enrich in conditions 3 and 4. That summarizes here region A. And you find that there are almost 5,000 hits across 47 different DELLs showing up. You then say, well, I'd like to make the thresholds more stringent. So you then can change the enrichment level from 10 to 100. You can lower this number from 10 to 1. And now you see that you have 2,000 potential hits showing up across 20 DELLs. Further, you can put in thresholds for things like heavy atom, C log P, and other things so that you can narrow it down and you can say how many potential hits fit the enrichment that I want to see in the conditions that I want with properties that I think would be useful as a potential hit. So a difference of the self-service kits and our normal full screening package, of course, is that you have to physically do this screen in your own lab, or you have to find someone else to do the screen for you. One of the things that we've done to help this along is that we have placed a learning lab in Boston. And just to show how serious about this we are, let me skip a slide for just a second. Here's a picture of the Boston Learning Lab, and it literally says right here, welcome to the DELL Open Teaching Lab, and welcome to the Delight Teaching Lab. So we mean it that you can show up here and you can learn how to do your screens. Now, that doesn't mean that you have to visit our lab to learn how to do these screens. We have shipped 346 kits. Many people have done the screen in their own lab without the use of our Boston Learning Lab. If you visit the portal that I showed you a couple slides back, between the schemes and the pictures, etc., you can watch these things step by step on how the screen is done. And I can assure you that as long as you watch and you learn, you can figure out how to do these DELL screens without visiting the Boston Lab. But if you can visit the Boston Lab, that is a great way to go. I also want to point out that the samples after your screen is done, you can ship them to Boston, you can ship them to Shanghai, or you can ship them within the EU, whatever works best for you, whatever's fastest, and whatever costs less. Okay, so real-time QC. So I did mention that that most screens work, that we've shipped 346 kits. A lot of people in different labs have done these screens, and we know that people can conduct these screens and that they work well. But I will say this, the Delight screens always work, almost. The DELL Open kits, depending upon the labs that they're shipped to, some of the DELL Open kit screens do really well. And there are other groups where the results of the DELL Open screens, they don't look that great. And part of the problem is that there's some QC issue going on during the physical screen. And so what we wanted to do here was to provide you with the primers and the reagents that you need so that you and your own lab now can do the QC work so that before you ship the samples back to us, you know that the selection went well. And then if the selection didn't go well, you now have the option of going back into the portal and getting a new DELL Open kit and just starting over, which might be the better choice. So let me talk a little bit about what these QC tests are. So the first problem that we often see is that somehow, for instance, DELL that is in tube 1 is somehow ending up in the results of tube 2. And we can see that that is what we're seeing here. We see the correct band, the correct blue band for this tube, but somehow we're also getting the results of the green band here. This is now a QC that you can do. None of us exactly know how this mixing of the different tubes happens. We think perhaps sometimes people reuse pipette tips, or sometimes they use the pipette tips in such a way that the sample sprays out, and we end up with DELL from one tube being mixed in with the results of the DELL from a different tube. But if these results look really bad, you're probably better off getting a new DELL Open kit, and the kits are free, and starting over. The second thing that we can do is we can determine how much DELL is actually coming out of each round of the screen. So depending upon the conditions of the screen, you would expect the amount of DELL that you would collect to be different. But the fact is, it almost always occurs within a certain range. And so if the amount of DELL that you collect is really low or extremely high, then probably something bad has happened. And once again, instead of wasting your time and money shipping the samples back to us, you're probably best off giving us a call. We can talk about this, and we might be able to determine that, again, the best thing would just be to start all over. Okay, so this is my last slide, and what I want to do once again is just to run through the difference between what this third generation DELL kit is and what the prior kits were. So first of all, we have billions of new structures which have never before been made and never before been screened that are now in these kits with a focus on trying to better cover drug-like space. The screen itself can be done by you and your lab, but now you have the tools to do the QC work so that when you send the samples back to us, you can feel much better about the fact that the screen was done in the correct way. After the sequencing is done and the data is ready to be viewed, instead of receiving a static PDF, you can now go online and you can interact with your actual data, and you can choose hits that fit the physical profile that you would like. And that can help you make the choice. Do you want to purchase the data set? Or in the case of DELL Open, which 5 hits do you want to choose and make and then test? Now, I want to talk about one other thing very briefly. Robert Abel will be speaking after me, so I won't dive into this in any depth. But one thing about DELL is that the amount of data that we produce is extremely large. And so the question has always been, can we look at all of this data and using AI or ML can we find a way to do a better job than a chemist would, deciding what hits to follow up? And can we take all of that data and also find a way to do virtual screens so that we could find other hits that might structurally look very different from what's showing up in the DELL screen, but might still be active? And so now with DELL Lite, you do have the option of, with an additional fee, of having the Delight kit run and then sharing the entire data set so that AI can then be used to possibly help you find more and better hits. And again, that's going to be discussed later. Okay, so with that, that was my last slide. I'm not terribly accustomed to these QR things, but I have found that if you take a picture of this, it will take you to a website. So it works. And with that, I'll hand it back over to Arsalan, and I'll be back for a Q&A at the very end of this. So if you have questions, I will answer them then.
All right, thank you, Alex. Now we're going to hear from several of WuXi's partners. And yes, don't worry, we are going to get to questions. There are tons of them in the queue. Keep them coming for Alex. I'm going to get to as many of them as possible at the end of our session. Now let's briefly hear from some WuXi partners. First up, we're going to have Dr. Jia Si, who is an assistant professor in the Department of Chemistry at the Miller School of Medicine and junior faculty at the Frost Institute of Chemistry and Molecular Science at the University of Miami. He's also a visiting investigator in the Department of Chemistry at Scripps Research in La Jolla. We're also going to hear from Dr. Erkan Balaglu, who is the founder and principal at Boston Biotech Consulting. And lastly, we will also hear from Dr. Robert Abel, the Executive Vice President of Science at Schrödinger, on his perspective of the WuXi and Schrödinger partnership. So let's hear these videos.
Hello everyone, I'm Jia from Scripps Research. I've been working with the DELL Open program for more than a year. Here I want to briefly share with you my experience and thought about the program. I think it's probably very similar to many other colleagues. When I first heard about this almost free DELL Open project, I could not believe it really exists, because at the same time other companies are charging hundreds of $1,000s for the service, and no one would share any of their libraries. I mean, which is understandable, because it is very expensive to make those libraries, and they indeed valuable assets of their companies. However, this situation makes DNA-encoded library technology almost inaccessible to people in academia, where actually the most innovations come from, right? So briefly, we got the comprehensive paperwork done, and we started to use the free libraries. So we were lucky to be the first batch of users for the distributed libraries. So we screened the libraries with several of our favorite targets. So I have to say that in the beginning, the process was slightly different from our previously oversimplified idea about how the screening should go. But the DELL Open team members gave us a lot of suggestions. We had quite a few times of effective and efficient discussions with them about the encountered problems. Later on, the bioinformatic analysis further provided more insights for the improvement. And with their great help, we were able to identify quite a few binders to our protein targets. I can see they are very rapidly responding to the feedback from the clients. The second version of DELL Open libraries came with a more detailed protocol, part of which was relevant to our increase. To me, the best part of DELL Open program, first, it gives free libraries and a free bioinformatic service. It gives you the information to decide whether to move or not to move forward. The compound synthesis service is also at a very reasonable price. The second part is confidentiality as well as the openness. And there was a very comprehensive paperwork signed up front protecting both parties. And there are multiple points that you can pause to choose whether or not to move forward and review your target. Right, so I would highly recommend people to try it out, and I wish the program can grow bigger and better, helping to promote basic and translational research from an angle never before. Thank you.
Erkan Baloğlu. I'm the founder and principal of Boston Biotech Consulting, a small boutique biotech company providing strategic, tactical, scientific, and operational expertise on all aspects of drug discovery and early clinical development. I'm a medicinal chemist by training with over 20 years of experience in the pharmaceutical industry, leading drug discovery programs and teams from early drug discovery through clinical candidate selection, preclinical development, IND filing, and early clinical development activities. As a scientist, I was aware of the DELL technology, but I never utilized it until last year. In my previous role as Vice President of Drug Discovery and Medicinal Chemistry at Restore Bio, where I was heading drug discovery operations, we decided to use the DELL technology to screen for and identify a novel hit for one of our programs. So after a detailed search and interview process of several service providers, we decided to collaborate with WuXi on this journey. And we were particularly impressed with the WuXi team, their depth of knowledge and experience in the science, their generosity in applying their expertise and advice on our program, the size of the compound library offered, and the state-of-the-art facilities equipped with the latest technology and instruments. And I know because I visited the Shanghai labs last year. Furthermore, we were attracted to all other services WuXi offered, such as syntheses, follow-up testing, and any additional studies that might be required. So basically, WuXi provided a very convenient one-stop shop for our small, all-virtual biotech company. I founded Boston Biotech Consulting earlier this year and started assisting my clients on their drug discovery and early clinical development programs. One of my clients, a university professor here in the US, reached out to me and asked for advice on how to initiate a drug discovery program on a novel target that they were planning to work on. I walked them through the phases of drug discovery and introduced several options in order to identify a hit as the starting point of their future drug discovery program. After weighing all options, reviewing the pros and cons of various technologies, it was decided to utilize DELL technology to identify a novel hit. And naturally, because of my previous experience and connections, we reached out to the WuXi team for a potential collaboration. To our surprise, we were offered the DELL Open option, a service provided to the academic community only. And most importantly, it was free of charge. So according to the offer, my client was required to practice the protein of interest or prepare the protein of interest. WuXi would ship the DELL Open kit. My client would then perform the screening in their labs according to the procedures and ship the materials back to WuXi for further processing. This was an amazing offer from several angles for my client because there is no commitment to pay any fees if no hits were identified. The protein of interest of the novel target could be kept confidential. All the expertise and the advice offered by WuXi scientists came free of charge. In addition, the potential to introduce certain level of selectivity early on at the seed identification stage made us feel confident that this was the best option for my client. So we agreed to proceed with DELL Open. WuXi also offered my client another great opportunity, the option to perform the screening at WuXi's all-new state-of-the-art facility in the Boston area, dedicated to DELL Open, with all the instruments and equipment needed, also free of charge. The protein was shipped to WuXi Labs, and all we had to do was to show up, basically. The kit and everything else we needed were waiting for us at WuXi's dedicated labs for DELL Open. After a brief introduction of the facility, we were given detailed procedures to perform the screening. Now, keep in mind, we had no prior experience on how to perform DELL screen. In addition, not only were the DELL screen procedures were extremely straightforward, but two highly experienced WuXi scientists were there with us, available to walk us through the procedures and answer any questions that we might have. The whole process was actually very pleasant. More importantly, we recently received the results of our screen and found out that several potential hits were identified and proposed for off-DNA synthesis. And WuXi is now also helping us with the syntheses of these hits. And my client and I can't wait to test these compounds to move this very interesting program to the next phase. How was that?
All right, thank you for that. And now I just want to remind the audience, we're getting so many great questions. We've got another short video here from Robert Abel coming right up, but please get your questions in, and I'm going to be asking them rapid fire to Alex here in just a moment. So thank you very much for all of that. We will now hear from Robert Abel.
Hi, I'm Robert Abel, Executive Vice President of Science at Schrödinger. I lead our computational science R&D portfolio, and I work closely with our drug discovery group, account managers, and application scientists to make sure our computational methods are used to greatest possible effect to advance our internal and collaborative drug discovery projects, as well as those of our software customers. Schrödinger's industry-leading computational science platform facilitates the research efforts of biopharmaceutical and industrial companies, academic institutions, and government laboratories worldwide. Our platform integrates best-in-class physics-based modeling with state-of-the-art machine learning and best-in-class enterprise-level solutions. We believe integrated use of these technologies can greatly increase the probability of success of drug discovery efforts and accelerate the realization of novel, more efficacious drug therapies. Schrödinger also pursues a number of wholly owned and collaborative drug discovery programs in a broad range of therapeutic areas. Our collaboration with WuXi will integrate Schrödinger's physics-based software platform and machine learning analysis with WuXi's DNA-encoded library service. We believe this will decrease the false positive and false negative rates of traditional DELL screening and will allow for more rapid identification of high-quality small molecule hits suitable for initiation of hit-to-lead and lead optimization efforts for a wide variety of medicinally relevant protein targets. We at Schrödinger are deeply excited to combine our machine learning and physics-based modeling capabilities with WuXi's ability to generate experimental data at scale. We further believe by leveraging the expertise of both companies in this strategic way, we will enhance our internal drug discovery efforts and will also be able to provide our partners and customers more and better avenues to discover new therapeutic candidate molecules. We at Schrödinger are very excited to be working with WuXi in this area.
Thank you. Lots and lots of questions. Cool video. Alex, let's get to the questions. There's several, so we have just under 15 minutes or so to get to them. So let me pose to you the first question here, Alex. How many samples can be screened by each 4 tubes of DELL?
So there's each tube can be used to screen a single protein sample. Each cell service kit has 4 tubes, but one of the conditions needs to be a tube where no target is being added. So that's the control sample. So you can screen 3 variations. So you could screen one target 3 different ways, or you could screen 3 different targets all at once, but only one condition per target.
Okay. I'm going to combine 2 of these questions together because I think I know the answer to one of them. But can industry use DELL Open? And can these kits be used on helicases or DNA binding proteins?
Oh, okay. So the answer for the first part is to use the DELL Open kit, you need to be an academic lab or a nonprofit. So, maybe the loophole here is you're a professor and you have a startup, but you also have your own academic lab. So you do have the choice then of using a DELL Open kit, running the screen in your lab with every intent of taking the hits that you find and using them in your startup. And that's perfectly fine. As far as the DNA binding protein, so the answer is yes, you can run a DELL screen on such targets. But the thing is, you need to determine if the DELL itself is binding to this target. And if it does, you need to find a way to block that binding from occurring. Probably the best way to do that is to add DNA, which binds to the target binding pocket and is going to completely block it. I think that that work could be done with a DELL kit. It could be done with a DELL Open kit. But it's I would consider it to be sort of more of an advanced user who's going to have to do that. I would say you're going to have a tough time being successful with that with a DELL Open or a DELL kit. But there are people out there who have experience in the DELL world, and now they're in the academic world, or then they are in the industrial world, and they could take on that task.
Okay. All right, now let's go to our next question. What support do you offer for assay development experiments? So for example, making and spiking in poscons to a background library to test enrichment.
Yeah, so if the goal here is to spike in a known ligand connected to a barcode, I would say that you're probably best off doing that with a full service screening package and not so much at this point in time with a DELL kit or with a DELL Open kit. Now, with that said, there's nothing stopping us or stopping you from taking the target and immobilizing it on a resin and then seeing if a fluorescent probe with your known binder is going to interact with the target. So as long as you know how to do those things, you're completely free to do those sort of tests, and you probably should, in order to make sure that your protein is still properly folded when it's immobilized on the resin. And that's work that you actually don't need us for. With all that said, I feel like the DELL and the DELL Open kits, if you've never done DELL screening in the past, these kits are really best for you if you keep it simple.
Wonderful. All right. Keep your questions coming. We have so many of them in the queue. So if we're not able to get to them, we'll endeavor to reach back out to you for some of these questions. But let me get to the next one over here. Have I well understood that we can send the samples after DELL screening to Munich instead of sending to Shanghai? This will enormously facilitate the process as it's pretty complicated sending compounds from Europe back to China. Can you please confirm?
Yes. So we are just now getting the Munich site ready to do DELL screens. It is 95% of the way there, and by the time we receive samples and Christmas is done, it will be completely ready. So yeah, you can now ship the samples to the Munich site.
Wonderful. Okay. Do you offer multiple selection technique protocols to choose from, or is there only one?
There's only one, really. So what we don't dictate is what the buffers are that you should use. So we do suggest a standard buffer, and if your protein is happy in that buffer, then it's great. But a nice thing about the DELL Open and the Delight kits is that we don't know how you've done this screen. And so you are allowed to invent and discover new ways to do these screens. So yeah, you can certainly use different buffers. You could use different temperatures. You could do all sorts of different things. The only thing that we say is when you veer away from the standard protocol that we offer, we no longer know what the results are going to look like.
Okay. Right. And now, is the readout limited to binding, or is activity also possible?
Yeah, so at the moment, DELL screens of this sort are only a binding assay. There are technologies to potentially do DELL screens that would be functional assays. It's not ready yet.
Okay. For data analysis, do you see for each library the preliminary SAR, like how different R1, R2, R3 groups contribute to enrichment?
Yeah. So especially with the Delight data set, when you unblind all those structures, you get this enormous data set sent to you. And you can visualize that data set, and you can often see what we call enrichment SAR. And so you can determine which BBs, which functional groups are important for the binding. With that said, determining SAR from the sequencing data can only be taken so far. In the end, the hits need to be synthesized and tested to really determine what the SAR is.
Okay. Alex, for 350 kits shipped, how many Delight kits in that batch? How many Delight customers order a follow-up kit?
So I actually know the answer to this, surprisingly. So yeah, there's 346 kits shipped. There's 64 of those were Delight kits. So a very large percentage were the Delight kits. And I think it's well over half of the Delight kits end up being purchased. So the full data package ends up being purchased. So once they see what the results are with the metadata, with the structures not shown, they decide to pay the fee and to see the structures. So that happens, I think it's 70% of the time the fee is paid.
Very good. Okay. Thanks, Alex. This is amazing. Can you tell something about the chemical space covered by the DELL Open Kit? Is it adapted for protein interactions, for example?
Yeah. So I like most chemists, we don't know exactly what is the best way to hit a PPI. Perhaps I would say the best way is to create structures that are physically larger, that maybe those have the best chance. Towards that end, we create DELLs that cover a lot of different chemical space. Some of it is smaller drug-like space, but also there's a very large chunk that covers chemical space that breaks the rule of 5. And perhaps that's where you want to be for certain types of PPIs. So I would say that we cover the chemical space well for both those groups, for the oral drug-like group and for the breaking the rule of 5 group.
I'm going to try to get to several more here in a quick fashion because we have about 4 minutes left. So here's a question. Great presentation. How do you pick the best 5 hits as the DELL Open Kit process?
So how this is going to be in the future with the 3rd generation kit is that you can go online, you can log into the portal, and you can interact with your own data set. And you're probably going to decide that you want to see things that enrich in one condition and that don't enrich in another. And then you'll narrow it down a little bit more about what the physical properties are. So I think a lot of that can probably be done on your own. reading the protocols for this that we provide to you, and then with you interacting with your own data. But in the end, if that process sort of breaks down, someone from the WuXi team will probably just be able to help you out.
Okay. Now, are the kits compatible with membrane protein targets and detergent?
Well, so in theory, yes. There's no publications showing this though, and I think this is one of those things where if you felt that you had a novel way to purify these membrane proteins and you had some ideas about how you wanted to do the screen, but of course you didn't want to make your own DELLs, then a DELL open kit might work well because of course you can get the kit for free and you can try it. The only thing that I would add is I would not have an unrealistic expectation of what the results are going to look like. It might work great, but you should not expect that.
Okay, let's try to get to two more questions here real quick. Can you comment on the requirements of protein target quality?
Yeah, so it's garbage in, garbage out for the most part. As a chemist, I like to blame the biologist for failures. So I would say when the screen doesn't work, it was probably the protein. Yeah, I mean, what more can I say? It's a binding assay. You need crystallography grade protein, or at least that's best. Now, with that said, I have run screens against protein, which wasn't that great. It wasn't very active, and it tended to degrade quickly over time, and we still found hits. You just have to take into account that when your protein isn't pure to start with, and when your protein isn't terribly stable, the risk of not finding good hits goes up.
Okay. All right, can you give us a success estimation for high affinity RNA to RNA binding protein for DELL open?
RNA to RNA binding?
RNA binding proteins.
Well, so if you're targeting an RNA binding protein, then I would say if it's stable and it has a binding pocket somewhere on it, then I would say your chances of finding it are really high, assuming that it doesn't bind to the DELL barcode itself. If your RNA binding protein has no small molecule binding pocket on it, then I think your chances of finding a hit decrease dramatically. So that's probably what it's going to depend on. Any protein with a nice binding pocket, you'll find hits for. And as that nice binding pocket becomes more and more shallow, your chances get lower and lower.
Okay, let's try to smuggle one more question in here, Alex, because I think it's a good one. If a hit is identified, the following synthesis, will it be done at WuXi or can it be partnership way?
So if a hit comes from DELL open, we don't share the structure with you, so we have to synthesize the hit for you. But we have a special cost for this which we have arranged with the academic group that oversees dellopen.org. For the Delight kit, however, once you purchase the data, the structures are there and you can do anything with that data set. So you could synthesize the hits yourselves, you could ask us to. You could go to some other group and have them make them for you. It's completely whatever you would like.
Okay, with that, we're going to have to conclude today's webinar. But thank you, Alex. It has been a true delight to work with you and the WuXi team on all this. Thank you for your presentation. Thank you to all of the panelists who presented today. I'm Arslan Arif for Endpoints News. Thank you for joining us, and we hope to see you at a future presentation. Thanks, everybody.